The topic is Overview of Development of Vaccine for COVID-19. The outbreak of coronavirus SARS-CoV-2 or novel coronavirus NCOV first reported to have occurred in Wuhan, China in the end of December 2019. The disease infected with NCOV was spreading across several countries beyond China because of its unprecedented infecting power. On February 11, 2020, World Health Organization WHO announced the name of the new coronavirus disease as COVID-19. COVID-19 had spread rapidly throughout China at high rate of transmission. Within February-March 2020, it caused panic to several other countries including Italy, Spain, United States of America, United Kingdom, and Germany. The outbreak was declared a public health emergency of international concern by WHO on January 30, 2020. Further, considering the devastation and deaths occurred in most countries, WHO on March 11, 2020, declared COVID-19 outbreak a global pandemic. The Director General WHO noted that over the past 22 weeks, the number of cases outside China increased 13-fold and the number of countries with cases increased 3-fold. Further increases were expected. He expressed deep concern towards the alarming level of spread and severity and for the alarming levels of inaction. He called on countries to take aggressive actions to contain the virus. Concern expressed by WHO is found to be infallible with progress of time and aggressive multiplication of the disease. Statistics as of now is horrifying. COVID-19 has so far affected 215 countries in the world. The disease, as on May 9, has infected over 4 million people globally and with death figure running to 276,000. In India, COVID claimed 59,000 infections and 1.8 thousand lives so far. During the fortnight, numbers of people infected increased by 1.5 times globally and 2.8 times in India. The affected countries, including India, invoked lockdown for about 2 months with likely extension for arresting the spread. The government, with expansive health services and civic administration, facing greatest challenge to contain the virus. Frequent washing with soaps and sanitizers, using masks and social distancing, as all promulgated in advisories, will keep some of us at bay from virus for only a brief period. All these awkward measures are not a permanent solution for the ongoing catastrophe. Pandemic with the lockdown wreaked havoc on the national and global economy. It has left millions of people jobless, MSME industry in shambles, and government exchequers in quandary. No specific method of cure from the disease and containment is on the anvil. All-round panic continues to engulf us. Development of vaccine against the virus is of paramount importance for saving the mankind. The vaccine is an agent that mimics the virus to set up B lymphocyte and T lymphocyte cells, but does not have the ability to overcome the immune system and cause the disease. The spike protein of the virus is a clove-shaped tumor and contains three segments: a large ectodomain, a single-pass transmembrane anchor, and a short intracellular tail. The ectodomain consists of a receptor binding subunit S1 and a membrane fusion subunit S2. The virus prefers to enter human body through nose, throat, eye, and finally to respiratory system and lungs. Its S1 binds to a receptor on the host cell surface for viral attachment, and S2 fuses the host and viral membranes, allowing viral genomes to enter host cells. Receptor binding and membrane fusion are the initial and critical steps in the coronavirus infection cycle. They also serve as primary targets for human invasion. Thus, virus merges with the body's host cells and multiply in large numbers within a short circle and infect the body. It destroys the body's immune system and may cause death. The immune system is made up of two armies of cells, innate and acquired. Innate immune cells are the body's first line of defense. They quickly respond to foreign cells to fight infection, battle with virus, and defend the body. Our acquired immunity, also called adaptive immunity, uses T cells and B cells when invading organisms. While T cells help destroy infected cells, B cells form antibodies that can keep the virus in check. In case of coronavirus, these are neutralizing antibodies that bind to the spike protein of the virus. These cells take longer to develop because their behaviors evolve from learned experiences. Adaptive immune cells remember foreign invaders after their first encounter and fight them off the next time they enter the body. This is the fundamental premise for how vaccines work. It uses a small, harmless amount of protein from a disease to allow the immune system to recognize that protein if the virus were to invade the body. Now we talk about development of vaccine, different methods. Globally, about 100 groups of researchers are working for development of the vaccine relentlessly. There are 4 main methods that they follow. First one, weakened virus method. The vaccine uses nCoV in weakened and inactivated form. Antigen presenting cells, APC, will bring about the T cells and B cells to defend the incoming virus from infecting and multiplying. The active features of weakened virus fails to infect the body. China has started to test this version in humans. Many existing vaccines like measles and polio were made in this method. The second method, viral vector method. By application of these methods of genetic engineering, some proteins of genetic information in the virus are deleted or altered. The virus is then engineered so that proteins that constitute its envelope resemble those of the target virus. The virus acts as a vector and transmits the relevant features to the human immune system, which develop antibodies against the target virus. The demerits of such method is that the immune system may act to block the vector itself. However, 25 groups of researchers are working to develop vaccine through this method. Third method, nucleic acid vaccine method. The genetic information of the target virus are put directly to body cells. The body cells create the proteins and surface features of virus, and the immune system creates the antibodies to combat the invading virus. This method had not been proven for combating any virus, and no vaccine had been developed earlier. However, 20 teams of researchers are working to develop vaccine in this method. The fourth method, protein subunit method. This method targets the coronavirus spike proteins or the domain that binds to the receptors of target cells. It is done by inserting proteins into the body cells along with added substances that stimulate the immune system. Another version of this kind uses virus-like similar particles that have no genetic content but are cells that possess just the coronavirus structure. This could be a very effective method, but such particles are extremely difficult to manufacture. 28 groups of researchers are on the job to find vaccine in this method. Now we come to the progress made so far as on 9th of May 2020. Italian scientists have claimed to develop a vaccine that has successfully generated antibodies in mice that work on human cells. The vaccine has been tested in the Spallanzani Hospital in Rome. It is said to be one of the most advanced stages of testing of a potential vaccine in the country, as the vaccine neutralizes the virus in the human cells. After single vaccination, the mice developed antibodies that can block the virus from infecting human cells. The researchers selected the two best candidates after observing that the 5 vaccine candidates generated a large number of antibodies. Israel on May 7 announced that the country had a remarkable breakthrough in the development of Covid-19 vaccine. Israel's Institute of Biological Research has developed a monoclonal neutralizing antibody which will effectively neutralize novel coronavirus in the body of the carriers. Oxford University, UK, initiated a phase 1 clinical trial of its viral vector vaccine on April 23, where 2 volunteers were injected, and Miss Eliza Granato is one of the first persons to get injected with the vaccine. Their vaccine was developed under 3 months by the university's Jenner Institute. It uses a weakened strain of common cold virus, adenovirus, that causes infections in chimpanzees. For the vaccine to work, scientists have taken the genetic material of novel coronavirus present on the surface of the virus and put it in the vaccine. The university hoped to get vaccine approved by September. Moderna, Massachusetts, USA, has been cleared by FDA for phase 2 trials, and the company will begin such trial with 600 participants very shortly. They are finalizing plans for a phase 3 trial as early as this summer. The firm would line it up for approval for public sale in the end of December 2020 or early January 2021. The Pune-based Serum Institute of India is the world's largest vaccine maker, and India produces 60% of the world's requirement. The institute have made a gallant effort by already starting manufacture of the vaccine following Oxford University's development in anticipation of final approval. Development of COVID-19 vaccine is in various phases of trials for guaranteeing the safety and efficacy. Subsequently, pilot-scale production and distribution will most likely push the time frame to the early quarters of 2021. However, Oxford University is optimistic of a positive outcome of the candidate vaccine and has planned to get millions of doses of the vaccine before the end of current year, even as results of the final phase of the trial are awaited. In retrospect, the dawn of convalescence from COVID-19 will be on our horizon. Let us, by all optimism, hope arrival of desired vaccine by the end of December 2020 or early January 2021. Thank you.